The upper levels of the human digestive tract, the stomach and duodenum, function as high-stress filtering systems, using extreme acidity and rapid flow rates to break down incoming materials. However, further down the tract, the environment shifts into a compartmentalized sequence of spatial gradients. The distal large intestine, or the deep lab, operates under stabilized, neutral conditions. In this deep lab, the laboratory facility drastically slows its transit time. This delayed pace provides the molecular technicians with the luxury of time required to execute complex organic synthesis without being rushed through the facility. Success in this sector depends on strict environmental standards. As the oxygen tension approaches zero, the chamber enters a state of anaerobiosis. This oxygen-depleted environment is mandatory. Even a small amount of atmospheric contamination can cause the delicate synthesis of life-sustaining vitamins to grind to a halt. Maintaining this pristine environment is a massive, coordinated workforce. The colonic sector hosts the highest density of specialized molecular technicians on Earth, exceeding 100 billion cells per gram of material. Operating at peak capacity, the distal large intestine functions as a hyper-sterile manufacturing floor, assembling biological tools that are fundamentally necessary for the host's survival. Every manufacturing floor requires a reliable loading dock. While the host's own enzymes process simple sugars and proteins in the upper lab, certain complex carbohydrates bypass early digestion entirely, arriving in the deep lab virtually untouched. These plant-based reagents, such as resistant starch and inulin, possess sturdy cell walls. Because the human body lacks the specific biological shredders required to dismantle these reagents, they serve as the primary fuel for the microbial workforce. Observe how the host's enzymes deflect off the reinforced geometric structure of these fibers. Specialized saccharolytic bacteria, the heavy machinery operators, possess the advanced enzymatic pathways to latch onto and dismantle these tough plant fibers, converting them into manageable bioactive molecules. A steady supply of these high-quality reagents acts as a boost in grant funding. As the reagent supply increases, it upregulates the proliferation of specialized lactic acid mepcuria and roseburia, significantly expanding the lab's capacity to synthesize complex coenzymes. Without these plant-derived substrates, the synthesis workstations sit idle, the reagents dry up, the workforce starves, and the laboratory's production line for essential nutrients reaches a full production halt. On the active production floor, master producers like Bifidobacterium and Bacteroides hold the genetic blueprints for manufacturing. These species contain the complete instruction manuals required to assemble active vitamins from basic organic precursors. To produce thiamine, specialized enzymes like Thi-I and Thi-4 coordinate the assembly, stitching carbon and nitrogen nodes into a complete molecule. This synthesized thiamine then docks into microbial pathways, powering metabolism and driving short-chain fatty acid production to keep the environment stable. These primary producers also export excess thiamine into local cross-feeding networks. This functions as an internal employee cafeteria, where prototrophic bacteria share the synthesized B1 with auxotrophic residents that lack their own synthesis blueprints but perform other vital laboratory tasks. Thiamine serves as both a primary output and the core power source for the facility. Without this continuous assembly, the collaborative exchange between specialized workers fails. Manufacturing is only half the battle. The intestinal laboratory must also use its chemical outputs to continuously maintain and repair its own physical structure. Microbially synthesized folate and cobalamin, B9 and B12, drive a master biochemical pathway called one-carbon metabolism. This pathway facilitates the DNA methylation required for the host's cells to differentiate and mature, providing the specific instructions needed to repair the intestinal walls and regenerate the lining. These instructions are vital for goblet cells. With robust B9 and B12, they successfully produce the protective moisture layer that coats the chamber. When depleted, the barrier thins and becomes discontinuous, leaving the epithelium exposed. Emergency expansions of the lining fail to restore the seal. Sustained internal vitamin synthesis provides the fundamental structural defense that prevents the reaction chamber's toxic contents from leaking into the host's systemic circulation. The collapse of the facility often begins at the loading dock. Metabolic shifts associated with obesity flood the lab with poor quality reagents, such as high-fat substrates and simple sugars, which favor the growth of unspecialized bacteria. This creates a budget crisis for the master chemists. As the specialized workforce starves, the lab falls into dysbiosis, a state of instability where opportunistic scavenging bacteria rapidly overgrow and prioritize their own survival over the production of vitamins. These opportunistic residents dismantle the unmaintained moisture barrier and fracture the epithelial tight junctions, the biological seals of the facility. This structural failure transforms the cleanroom into a critically compromised, leaky lab. As the walls fracture, a biohazard spill occurs. These jacket toxins, known as lipopolysaccharides, escape the reaction chamber and enter the surrounding environment, triggering the facility's emergency alarm system. The host responds to this inflammatory spill with a biological lockdown. Pro-inflammatory markers act as a roadblock, damaging the transport proteins that serve as the conveyor belts for nutrients. To contain the inflammation, the host effectively seals the exit doors, preventing any remaining vitamins from being absorbed. When the laboratory's clean room conditions are compromised, the system reaches a state of functional paralysis. The walls are broken, the workforce is depleted, and the host's own defense mechanisms prevent the delivery of life-sustaining vitamins to the rest of the body.